Hey there, science fans! Today we're diving into the exciting world of biotechnology and genetic engineering. These fields are changing how we approach health, agriculture, and even what it means to be human. At the heart of this revolution is a groundbreaking tool called CRISPR. CRISPR is like a tiny pair of scissors that allows scientists to edit genes with incredible precision. Imagine the possibilities. We could potentially cure genetic diseases, develop new treatments, and even enhance the resilience of our crops. But with great power comes great responsibility. As we explore the potential of genetic engineering, we must also carefully consider the ethical implications. Is it right to alter the very building blocks of life? What are the risks involved? These are just some of the questions we'll be tackling today. So buckle up and get ready for an incredible journey into the future of biotechnology. So, what exactly is CRISPR and how does it work? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a mouthful, I know, but basically it's a defense system that bacteria have been using for millions of years to fight off viruses. Think of it like this. Bacteria store little snippets of viral DNA in their own genetic code. Then, when a virus attacks, the bacteria can use these snippets to recognize and cut up the virus's DNA, stopping the infection in its tracks. Pretty cool, right? Scientists have figured out how to harness this natural system to edit genes in any organism, including humans. Using CRISPR, we can target specific genes, cut them out, replace them, or even just make small changes to their code. This technology is revolutionizing how we study and treat diseases, develop new crops, and even understand our own evolution. The potential applications of gene editing are truly mind-boggling. Let's start with healthcare. Imagine a world where we could cure genetic diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease. These diseases are caused by errors in our DNA, and CRISPR gives us the power to potentially correct those errors. But it doesn't stop there. Gene editing could also lead to new treatments for cancer, Alzheimer's disease, and HIV/AIDS. By targeting the genes involved in these diseases, we could develop more effective therapies and potentially even prevent these conditions from developing in the first place. Beyond healthcare, gene editing holds immense promise for agriculture. We could engineer crops that are more resistant to pests, diseases, and harsh environmental conditions. This could help us increase food production and feed a growing global population. And by reducing the need for pesticides and herbicides, we could also minimize our impact on the environment. Now that's a win-win. You might be thinking, this all sounds great, Bill, but is any of this actually happening? The answer is a resounding yes. Gene editing is already being used in labs and clinics around the world. In agriculture, scientists have used CRISPR to create mushrooms that don't brown as quickly, tomatoes with increased shelf life, and even soybeans with healthier fats. These genetically modified crops are already being grown and sold in some countries. In medicine, CRISPR is being investigated in clinical trials for the treatment of various diseases, including cancer, blood disorders, and inherited blindness. While still in the early stages, these trials offer hope for patients and their families who have been battling these devastating diseases. And it's not just humans and plants benefiting from this technology. Gene editing is being used to develop new treatments for animals too. For example, researchers are exploring the use of CRISPR to create disease-resistant livestock and even to bring back extinct species like the woolly mammoth. Now, wouldn't that be something? Section 5. The ethical frontier, weighing the moral implications. As with any powerful technology, gene editing raises ethical concerns that we need to address carefully. One of the biggest concerns is the potential for unintended consequences. When we edit genes, we're making changes to the very blueprint of life. These changes can be passed down to future generations, so it's crucial to consider the long-term effects of our actions. Another ethical dilemma is the issue of designer babies. Imagine a world where parents could choose their child's eye color, height, and even intelligence. Is this something we want? Where do we draw the line between medical necessity and enhancement? These are tough questions with no easy answers. Furthermore, there's the question of access and equity. Gene editing technologies are expensive, and there's a risk that they could exacerbate existing inequalities. We need to ensure that these technologies are available to everyone, not just the wealthy and privileged. It's a matter of social justice. Section 6, Case Studies examining CRISPR's impact. Let's look at some real-world case studies to illustrate both the promise and the peril of gene editing. In 2018, a Chinese scientist shocked the world when he announced the birth of twin girls whose genes he had edited using CRISPR. His goal was to make the girls resistant to HIV, but the scientific community condemned his actions as unethical and potentially dangerous. On a more positive note, in 2019, a young woman in the United States became the first person to be treated for sickle cell disease using CRISPR gene editing. The treatment involved removing some of her bone marrow cells, editing the faulty gene in those cells, and then transplanting the modified cells back into her body. The results were remarkable. Her symptoms disappeared and she no longer needed blood transfusions. These case studies highlight the complex ethical and societal implications of gene editing. They also underscore the need for careful regulation and oversight to ensure this technology is used responsibly and ethically. Section 7, Looking Ahead, the future of gene editing and our genes. The future of gene editing is full of both promise and uncertainty. As technology continues to advance, we can expect to see even more innovative applications in healthcare, agriculture, and other fields. We might even one day be able to use gene editing to enhance our own abilities, like our intelligence or physical strength. However, it's crucial that we proceed with caution and responsibility. We need to have open and honest conversations about the ethical implications of gene editing and establish clear guidelines for its use. The decisions we make today will have a profound impact on the future of humanity and the planet. It's an exciting time to be alive, folks. We're on the cusp of a new era in biotechnology, one that has the potential to solve some of the world's most pressing problems. But we must remember that with great power comes great responsibility. Let's use this powerful tool wisely to create a better future for all. Section 8, Conclusion, Balancing Promise and Peril in the Age of Genetic Engineering. We've covered a lot of ground today, from the basics of CRISPR to the ethical dilemmas of gene editing. I hope you've gained a better understanding of this revolutionary technology and its potential to reshape our world. As we move forward, it's essential to remember that genetic engineering is a double-edged sword. It holds immense promise for curing diseases, improving agriculture, and even enhancing our own abilities. But it also raises profound ethical questions about our relationship with nature, the future of humanity, and the very definition of what it means to be human. So what do you think? Is gene editing a force for good or a slippery slope to a dystopian future? Let's keep the conversation going and work together to ensure that this powerful technology is used wisely for the benefit of all. Remember, the future is in our hands.